Alright, we're going to look in chapter 12. In the case of gene cloning, what we're going to be doing is taking a gene from a cell, like a eukaryotic cell we have over here, and we're going to extract that gene and then add it to a bacterial plasmid. And that's going to create what are called recombinant plasmids, and we'll see how that's done here in a bit. So what we'll typically do is we will isolate a plasmid from a bacterium. Usually the bacterium is going to be E. coli, and it's going to really serve as kind of our vector, or it's going to be essentially our gene. Now, what we do is, once again, is we will cut the gene from the cell that we want the gene from. So this is our gene of interest. And what we're going to do with this gene of interest is we're going to, like I said before, insert it into a bacterial plasmid. Now the way we're going to do that is we're going to use an enzyme to make a cut in the plasmid. Now once we've cut the plasmid with that enzyme, we'll use that same kind of enzyme to make a cut for this gene of interest that we have. Now once we get those cuts made, now we can get the gene of interest. We've cut it out. Now we have a cut in the plasmid, and we can get that gene of interest inserted into the cut plasmid. So now we get the target fragment, the gene of interest, and the plasmid. We get them combined, and actually we use an enzyme called DNA ligase to help join all this together. So now what we end up with is we have this plasmid with this gene of interest inserted and tied together. So we now have this recombinant DNA plasmid. Now what we'll do is we'll get bacterial cells. We'll mix this plasmid with some bacterial cells and get them to take up this plasmid by a process called transformation. And then what that bacterium will do is it will start to reproduce. Now when it reproduces, of course, it's reproducing this plasmid and this gene of interest. And then what we can do is we can that gene of interest will be transcribed and translated by those bacterial cells and produce the protein that we're after. And then we can harvest the protein. And that's one of the ways in which we get stonewashed genes, is by this process. Some of the proteins can actually be used to dissolve blood clots in heart attack therapy. Genes can also be inserted into other organisms. For instance, a gene can be used to alter bacteria for the cleaning up of toxic waste. And then genes have also been inserted into plants to confer. Now, when we talk about genes being moved into other organisms, cross-species lines, we're talking about GMOs, genetically modified organisms. And so what we really have is we have recombinant cells, recombinant organisms, and oftentimes what we get out of this, we get mass produced gene products. So for instance, if we look in our table 12.6, human insulin is made by E. coli, and it's used in the treatment of type 1, type 2 diabetes. So you can see E. coli is used to produce not only human insulin, but human growth hormone and other signal molecules. And so GMOs are actually not terribly uncommon. And E. coli is actually kind of the best organism whenever you need, in many cases, when you need just a protein being produced. And the thing about E. coli is that they grow in a hurry. And it's pretty inexpensive to keep them and culture them. And they can produce lots of different kinds of proteins and in large amounts. And it's pretty easy to extract the protein products because what they'll do is they secrete their protein products directly into their growth medium. So it's pretty easy to get a hold of those protein products. Now, Saccharomyces cerevisiae, which you think of as being just it's yeast, it's a fungus, but anyway, it's oftentimes the choice organism for when you need to produce a protein that involves really using a eukaryotic cell. Like I said before, it is a fungus. It's actually yeast. Saccharomyces is actually yeast that we use in making bread and beer, and it's pretty good at taking up foreign DNA and incorporating it into its genome, kind of like what we saw with E. coli on a previous slide. But oftentimes it's better at synthesizing and secreting eukaryotic proteins than E. coli, and there's a reason for it, because number one, Saccharomyces is a eukaryotic organism, and it has all the essential metabolic machinery to do that, to be able to produce these eukaryotic types of proteins, and some of the changes and modifications that need to be made to them, it can do pretty well. So the hepatitis B vaccine, which contains a hepatitis virus envelope protein, oftentimes is produced there by Saccharomyces cerevisiae. Now, mammalian cells are oftentimes the choice for making glycoproteins, and this is because in order to get the sugar attached to the protein, that's what glycoprotein, glyco means sugar, but to get that sugar attached to the protein the right way, then you need the right kind of machinery. Now there's something called EPO, and it's been in the news lately as well, specifically with Lance Armstrong and Tour de France and blood doping and things like that. But EPO is a hormone, and it stimulates the production of red blood cells, and it's really important though for individuals as they're being treated for anemia. It can be certainly a life-saving Now there's been ongoing research by pharmaceutical researchers as they've been trying to figure out better ways of mass production of certain gene products, and they've been looking at whole animals and whole plants instead of just culturing cells. And one of the things they show here in the bottom right hand picture anyways, you see this goat, and the goat has been modified in a way to where the milk it produces contains antithrombin. Now, antithrombin is a human protein, and it helps prevent inappropriate blood clotting. So it can be this protein can be isolated from the milk, and it can be used in reducing the risk of blood clots that can occur either during surgery or childbirth or something like that. They've also been able to develop sheep that produce milk, which contains a human protein called AAT. Now this protein is normally made by the liver, and white blood cells produce an enzyme that's critical to fighting infection. And AAT stops the white blood cell enzyme from attacking normal tissues, especially in the lung and liver. And so some individuals, some patients who have a hereditary form of emphysema can receive this treatment. So once again, an example of mass production of gene product using a whole animal instead of just a batch of cultured cells, essentially. So GMOs, the real downside to them is that they're difficult to produce, and well, DNA technology though has really changed the pharmaceutical industry and also medicine. It wasn't that long ago that insulin was actually used from pigs, but today insulin, of course, which is so critical to our ability to be able to regulate blood sugar, blood glucose levels, is produced by our pancreas. But there are some individuals who develop type 1 diabetes. They kill off their beta cells of their pancreas so they can no longer produce insulin. So, you know, prior to 1982, most insulin came from slaughtered pigs and cattle. And the insulin that they would extract from the pigs and cattle is not identical to human insulin. And some people would actually have allergic reactions because the protein, the insulin protein, is a little bit different. So in time, 1982, you had humulin produced by bacteria. It was the first recombinant drug approved by the FDA, and that was in 1982, and it's benefited a lot of people, continues to do so today. Now, HGH, human growth hormone, is another hormone. There are other animal growth hormones, but they have a little different shape. They're not as effective with humans. Prior to the 1980s, HGH was actually extracted from the pituitary of cadavers, but there was a real drawback to that because in 1985 it was discovered that this could actually lead to the development of Creutzfeldt-Jakob's disease if the HGH was extracted from someone who had had the disease. So in time, in 1985, artificial HGH gene was constructed and was joined with some human DNA. And now E. coli actually produce HGH, and this can be used then to treat children who are born with a particular form of dwarfism to where they'll develop more normally. So once again, these GMOs can certainly provide treatment to some individuals that are suffering different types of. Now, DNA technology has also aided the production of vaccines, because in vaccines oftentimes what we have is we have a harmless variant, a mutant of the antigen, or even it could be a dead antigen, or it could be a derivative of a pathogen. Now in the case of the mumps virus, what is used is an attenuated, in other words a live mutant mumps virus is used in the vaccine for vaccinating against mumps. And what happens is the process is one that involves inoculating another host to where some harmless variants, harmless strains that have lost the ability to infect human cells is produced. And in that way, that can be given to individuals to inoculate, prevent them from getting, once they're up on exposure of the mumps virus, developing mumps. The smallpox vaccine also contains a harmless variant as well, and the polio virus is a dead virus. Now, hepatitis B contains just the surface proteins of the virus. Like I said before, is actually made by yeast, Saccharomyces. So once again, DNA technology, the understanding of DNA technology, the growth in DNA technology has really made. Now GMOs, and GMO stands for genetically modified organisms. They're finding their way certainly into agriculture. They have for quite some time. Now, people in the past would certainly selectively breed crops to improve the nutrition level of the crops. And the difference between, say, that and GMOs is that in GMOs you actually have the insertion of genes in an artificial way. So it could be that the gene could even be from a different species. Now, if that's the case, then the recombinant organism is called a transgenic organism because it's across species lines essentially. So there can be, for instance, in the case of agriculture, a gene from one species being inserted into the plant, and that confers the plant with a new trait, which could mean that it's more resistant to a certain type of fungal infection, or it may encourage its growth rate or its ability to tolerate lower temperatures, all kinds of new traits. Now, this involves kind of what we described before, and that is getting the gene into, say, a plasmid, getting that plasmid then to actually make its way into a plant cell, and then that way the gene can get into one of the plant's chromosomes, and in doing so, now the plant can has this new trait, this new beneficial trait that it didn't have before. Well, of course, when you think about agriculture and the possibilities there in terms of some of these transgenic plants being able to grow and it may be more saline soils or without as much water, when you think too that the estimate is somewhere around 1 billion people facing malnutrition across the planet, GMOs maybe perhaps they're part of the solution. GMOs have improved food production, have improved shelf life and pest resistance, and also even the nutritional value of food stuff. Now, one example of this has to do with papaya ring spot. Well, papaya ring spot is caused by a virus, and it started making its way through Hawaii's papaya industry. And scientists at the University of Hawaii, they were able to genetically engineer a papaya ring spot virus resistant strain of papaya. So that way, that pretty much kind of stopped the viral infections. So now there were some, though, that didn't like the idea of these genetically engineered papaya plants. And so there were occasions when literally thousands of papaya trees on the big island of Hawaii were actually hacked down under the cover of darkness. And what most assume was going on there was it was a protest against the production of these GMO papaya plants. Now, in India, there was the insertion of a natural gene, but it's a rare salinity resistant gene, and this has actually enabled rice growing to occur in water that's 3 times as saline, 3 times as salty as seawater. And so this has allowed the growing of food in some areas where they couldn't grow the rice before. So that's an example, another example of DNA technology enabling more people to have access to more food. Now, golden rice is another agricultural crop that is a transgenic crop, and it contains daffodil genes. Now, what it can do is it can actually it's a rice that can actually produce beta-carotene. Now, beta-carotene is critical in helping prevent blindness in some parts of the world. They actually came up with what's called a golden rice 2, and it uses corn genes to actually boost the beta-carotene levels to even higher concentration levels. So there's all kinds of examples of GMOs. In 2006, researchers they genetically modified pigs to where they could carry this roundworm gene, so that as the pigs were putting on tissue, this roundworm gene's protein would actually convert some fatty acids into omega-3 fatty acids. And so the transgenic pig actually contains 4 to 5 times more omega-3 as a regular pig. They've done this with salmon as well. They've modified salmon to have a more active promoter of a growth hormone gene from Chinook salmon. And so that way they'll actually mature in less than half the time. But there are certainly some who are concerned about GMOs and about their safety. So there was a study that was done of the vitamin A production from the golden rice that we just mentioned a bit ago, and they also compared the percentage of absorbed and converted beta-carotene to vitamin A between golden rice, spinach, and a capsule of pure beta-carotene. And this was published back in 2012. So 68 Chinese school children were fed golden rice, which contains, the higher levels of, or the gene for beta-carotene production, spinach, which is a natural source of beta-carotene, and then there were some of the school children that actually received a capsule of pure beta-carotene. After 21 days, or during the 21-day study, blood samples were drawn to measure how much vitamin A the body was actually producing. Now, you can look at the graph below, and what you can see is that there's not much difference between the golden rice and the capsule, the children that were eating golden rice and the children that were given a capsule of pure beta-carotene, a capsule of the pure beta-carotene. But each of these were significantly more than what they were getting, say, from spinach. So there was a real benefit, certainly, in vitamin A production with the golden. And perhaps someday gene therapy may help us treat even more kinds of diseases as well. because we may be able to use viruses to infect human cells, to insert genes that are defective and cure diseases. So what about transgenic humans one of these days? That's the idea. That would be the result of gene therapy in humans. You could potentially take a gene from a healthy person. You could get that gene into an RNA version. You could get that RNA version into a retrovirus. Then you could remove bone marrow cells from a patient and infect those cells with this virus. The virus would insert a DNA inversion of its genome, including that normal human gene, into the cells, and then you could get those cells to go ahead and grow, and then inject them back into the patient, and now the patient is cured. The patient can produce what protein it really needs to. Something like this was tried with skids from 2000 till 2011. Gene therapy had cured 22 children with severe combined immunodeficiency, skids. There were, however, 4 of the participants that ended up developing leukemia, and that's because this gene was inserted near an oncogene, and so there was overproduction and out-of-control cell growth. One individual did die after the inserted gene turned some of the blood cells cancerous. And so, of course, what goes on with these individuals is that they don't develop a normal immune system. They can't develop an immune system. And so if they're not developing functional, say, T lymphocytes, which are really the critical members of adaptive immunity, then they quickly die from infections. And so some of these individuals, they have to live out a life within kind of a protective bubble. And whenever they do bone marrow transplants, it's only effective about 60% of the time, and that's a fairly. Well, in 2009, there was a gene therapy trial that involved LCA. Now, what happens in the case of LCA, or one form anyway, is that it has to do with a development of abnormal rhodopsin. And rhodopsin is a pigment, and it allows or enables the eye to detect light. And what happens with individuals with this form of LCA is that their photoreceptor cells gradually die, and so they become progressively blind. Now, in this trial, they injected a patient with a single injection containing a virus that was carrying a normal gene that would improve the vision in the eye. Now, it worked to some degree where it did allow, in some cases, some normal functioning. So once again, another example of some early work with getting some normal genes into humans and fixing some of the diseases. Now the question that comes up is, well, if is this a slippery slope? Will doing this kind of thing lead to eugenics, where you're trying to control the genetic makeup of human populations? And if that's the case, where would it end? What kinds of problems would be encountered, and how would we deal with those problems effectively?